Good afternoon, everyone. So remember, today is a review session, so obviously I'm going to be kind of expecting y'all to have questions, otherwise we'll just end a little early, okay? So just wanted to go through our typical reminders. Remember that quiz 7 is due on Sunday at 11:59 as always, but I really recommend that you take it before you take the exam. It'll give you an excuse to practice some of these questions, and even though I can't reveal the correct answers, like even the ones that you got wrong, you can at least use that to get pretty close to the ballpark of knowing, okay, I at least got these 13 out of 15 right. I know I'm good on those. These are the 2 that I need to figure out. Pretty straightforward. So please use that opportunity to basically study for your exam. Exam 2 is on Friday. Remember, you have 2 options to take it. Well, technically 3. For those of you that are SAS, you need to schedule with them and make sure you have plenty of space for that. If you need a Scantron, come see me just in case they don't have one for you, because I just sent them via email and I'm not sure if they give you one or not, but they should. And if they don't, come talk to me. I'll make sure it gets fixed. The other 2 options for that the majority of you really have are you can either come in here and take the test in person. And mind you, I only printed off about 100 copies. So if you haven't let me know, now would be the time. However, since it seems like a majority of y'all wanted to have this opportunity to take the exam online, what I've done is using the Respondus browser, which you guys can download from the test description that's available on web courses, as well as I think UCF has a site page that explains everything you need to know about it. The big point here is that you take it online. You have from 8 a.m. till 3 o'clock in the afternoon. That's as big of a gap as I could keep it open per what the department told me. So it shouldn't be that. It's the exact same questions with the exact same time limit and everything. So it's not like you're getting any benefits or drawbacks other than I like to take my tests in person or I like to take my tests using Respondus with the webcam. So just keep all that stuff in mind. Again, if you want to guarantee that you get a spot for the in-person stuff, send me an email. All right. Before we get kind of going into the actual review question, does anybody have any more like more procedural kind of questions, like anything issues with how you want to take the exam, anything like that? Yes.
If I've never done it, the Respondus thing, how would I go about
So to download Respondus, if you open up the assignment on web courses, right, like you would for opening up a quiz or something, there's that description right at the top there, right, that explains this is what you're doing, all that kind of fun stuff. There's a link in there that will take you to downloading the Respondus browser. That's all you need to worry about. And if it's not there, shoot me an email. I'll make sure it's there. Yes.
Is there anything for Respondus, like
So that is a good point to bring up. With Respondus, you do need a webcam that's enabled on your computer. And I'll be honest with you, it doesn't seem to work really, or it doesn't seem to agree really well with any of you that are using like a Chromebook. Because there's something about when it's not on Microsoft or Apple, it just doesn't agree quite right. Keep in mind too, you have the library as an option to take it at, both on their computers as well as just having a quiet space to take it. That a lot of them do have webcams because of how life has been the last year or so. But if you're having any computer issues, just let me know immediately. If I know that you are having computer issues immediately, I can usually fix it. If you let me know after that exam window is closed, it makes it a lot harder for me to fix things. And keep in mind too, there are still excused absences for this. Like say for instance, if you're at a funeral or if you have something that happens immediately that day, you can miss it for an excused reason. However, make sure 1, it's a true excuse reason, and 2, that you let me know as soon as possible. Because if you tell me 2 weeks later from Friday after all the grades are in, it makes my options a lot more restrictive. And while I want to give you all the benefit of the doubt as much as possible as I can, there's some things that the biology department says that I can do, and there's some things they tell me I can't. So remember that if you want the benefit of the doubt and making sure everything is perfect for you, if something happens, tell me it almost immediately, if not at least the day after. All right? Yes. Okay, so there are exactly 40 questions. You'll get roughly about a minute and a half per. which we did 35 last time and everybody finished before that 15 minutes was up. So it's 5 extra questions. And I have a reasoning for that. And that's usually because, especially with this test, it gives me the ability to add a couple more filler questions that are a little bit easier to answer. So for instance, like I can throw stuff about indirect versus direct development, some less like, did you memorize exactly what this term means questions, and gives you a little bit more buffer room. So even though it's a little bit more questions, it's not that they're easier, they're still relevant and exactly what you need for this exam, but they're just not as memory recall heavy, if you will. Any other questions? Yes.
Is it like quiz 4 to 7 or?
Yeah, so anything that happened since we finished our last exam is on this test. So including the lecture that was on Monday, which means for those of y'all that have missed lectures recently, you may want to go back and make sure you actually watch those videos. I see the watch times on them. They're only like 30 to 40 people actually watching them, and there's 450 people in here. And there's definitely not been that many during the lectures that I've seen. So just keep an eye on that. I know it seems tedious, but at least watch them. I know they're not the best quality, but it's better than nothing. And there's going to be things that I say that may not necessarily be on the slide. I know a couple of y'all ran into that buzzsaw with the direct versus indirect development, because even though I've said it 4 or 5 times talking about like these different situations with things outside of humans, it's not always written up on the slide. All right. Any other questions? Oh, yes.
How specific is the exam going to get in terms of the immune system? Like, are we going to need to know like tomorrow?
So with the immune system specifically, I'm going to keep it very broad. I'm not going to have you know every single little detail about a B cell or a T cell or anything like that, but I do want you to know the general categories. Like, you don't need to know how that whole system works very well, but you should know what a B cell is, what a T cell is, that kind of thing. Any other? Oh, sorry.
How about, like, it's kind of the same question, except for like brain, like hormones, like how specific are we getting in that?
The way I'll put it is, if there's an entire slide devoted to it, it's probably fair game. And honestly, keep in mind too, you've probably seen all of the questions that you're going to see on the test in either the way it is now or in rewritten form through all these quizzes. So keep that in mind. Yes. No, there is no difference at all. Yes.
My question's for Jada and for Bennett.
Mhm.
So this morning, I was like hearing a bird and I whipped out my camera and took a video.
Yeah.
And I tried to upload it, but I could only upload either a picture or the recording.
That's unfortunately one of the hangups that I've had with iNaturalist. One thing they haven't quite figured out yet, the audio recording thing has only been since the last 6 months.
Oh, okay.
So it's just because they didn't have that much storage space for their servers.
For sure.
So obviously, like, there's a big difference between a single picture and like a 30-second long video. It's a huge deal as far as like how much storage space there is. But yeah, unfortunately, you either have to record the sound with their stupid app, which can be problematic. But
Yeah.
yes. Anything that's happened since we took the last exam. Yes. It usually, at least from the students that I was interacting with for the previous makeup exam, it doesn't seem to agree with Chromebooks very well. I'll leave that decision up to you. I mean, there are, computers at the library as well, but keep in mind that I've seen people get it to work, but what I would suggest is maybe double check it and make sure it's working before you try to take it. And if something happens where like you start an attempt and you don't, and it runs into issues, just stop answering questions from that point or something like that. Even if you can't even get to the point where you actually are taking questions, I could at least reopen it and be like, oh yeah, you clearly didn't actually finish the test. There was something that happened and I can fix that, Yes.
Just that, for the class, I have a lot of questions here. I would have to use the first time I downloaded it, it gave me all kinds of trouble, but if you make it
Mhm.
30 seconds.
Yeah, it can be a little problematic at first, especially, but Keep in mind too, also in that test description that I mentioned that has like a link to Respondus, I have the phone number and the email for IT. So if something happens and shit hits the fan, don't be afraid to let them know and literally CC me in it, and I'm happy to like jump in and help however I can. Yes.
Figure 8 of the, like, through the heart.
Yeah, we can talk about that stuff. So what I'll do first is we've got a couple questions that people have already asked, and then we can jump back and explain some of that stuff too. All right. Is everybody else good on just like the procedural stuff, like how the test is going to happen, the extra credit, all that kind of fun stuff? Yes.
How many questions are there?
40. All right, so let's go ahead and get started in the action. Yes.
What's like the breakdown wise, like how much of the test comes from each section?
There's exactly the same amount per section. Yeah. I went through a lot of trouble making sure that, and that's just because I hate it when you take a test and it's like really weighted towards like a single lecture that you may have missed or something like that. I try to keep it very equitable. Right. Any last second questions? Okay. Let's get to the actual review portion here. So each one of these is based on, each one of these things I'm going to talk about was based off of somebody that had an issue with a previous question that either couldn't be here today or didn't want to talk about it in person. So the first thing I want to start with is I know y'all hate that stupid roots and shoots question, but it's a really simple question if you just totally ignore that first part of that sentence and look at a shoot does blank or a root does blank. That's all you need to focus on in that question, right? So in that case, you have these two different systems in plants. You have the shoot system, which is going to be the above ground portion. And that's going to be where it's going to have the stems to support leaves, which are obviously going to be there to carry out photosynthesis, i.e. producing sugar for that plant to survive. As well as you're going to have that vascular system, which is going to move sugars throughout the plant. Which is where the root system is there to anchor that plant in the soil, which some plants have very well-developed root systems that are going to hold them there a lot better than others. Some are, designed for hurricane force winds. Others could be knocked over in a breeze. And ultimately, what that root system is there to do is to gather up water and minerals that are present in that soil, move it into that root system, and then transport it up to the top where they might be needed for other things. So hopefully that made it a little bit more clear. Honestly, that question is tricky. Just make a point to take a second and read it carefully, and it will make a lot more sense. So I wanted to put this up here because everybody kind of, or not everybody, there was about 60% of people got it right. But when we were talking about synapses, and remember, a synapse is that junction, that space between a nerve cell and whatever cell it's communicating with, whether that be another nerve cell or a muscle cell or what have you. The synapse is simply this junction here. Yeah, you have the synaptic cleft, you have all these specialized things that are associated with it, but the synapse isn't actually what's sending out the signal. It's that space in between, it's the railroad tracks, it's not the train. Hopefully that makes a little bit more sense. So ultimately, remember, the thing that actually sends out the signal itself is the axon. So here you have the bottom of the axon coming out here. It's going to have all these little spots here where it's sending information out, sending out those neurotransmitters into that synapse, which will then interact with the other things. Yes.
So the axon sends the signal to the synaptic cleft,
Exactly.
which then causes synaptic vesicles to leave the synaptic cleft into the synapse.
Yes, into the synapse itself. So basically what's happening is you've got this opening right here, it's called the synapse, right? And the synaptic cleft is part of the synapse. And basically what's happening is you have this axon that's moving through all of that action potential. It's going to get down here. It's going to tell it to send the bottom part of the axon here to then release neurotransmitters into the synapse itself. And then the receiving cell is going to pick up those neurotransmitters. Basically, the neurotransmitters are moving through that synapse. The synapse is just kind of like a trap for them, a place where they can move and not get damaged. Does that make a little bit more sense?
So the bit that expands at the end is not the synapse.
No.
It's just the area that is also currently labeled as synaptic cleft.
Yes. So this part with synapse here is called the synaptic cleft. Synapse is just kind of interchangeable depending on what you're talking about in the circumstance here. So these little openings here are just the end of the axon. They're not actually like a separate thing. Yes,
Just to be sure the synapse and the synaptic cleft are the same thing, it's just the spacing.
exactly. Yeah, it's basically the same exact thing. Unfortunately, a lot of times in biology, you'll find that like based off of if you're talking about it singularly or plural, all that kind of fun stuff, it just kind of depends. But yeah, ultimately a synapse is just that space in between. All right. So the next thing I wanted to brush back up on are the two different control feedback loops that exist. You have the positive control feedback and the negative control feedback loop. And what this means is there's kind of two different things that happen. A positive feedback loop will continuously build upon itself. So if something releases some sort of endocrine or hormone or whatever, that hormone will trigger something else, which triggers just increased amounts. It never either slows down or it never like levels off. It always will perpetually move to, it's kind of like, do you guys remember when we talked about entropy? Where it's like things always are going to become more and more chaotic. That's what a positive feedback loop is, essentially. It's progressing more and more, building bigger and bigger until ultimately there's nothing left. It's out of resources, it can't react anymore. Whereas with this negative feedback loop, it's like your air conditioner, or really just like the thermostat in your house, right? You have something that receives a stimulus, so either you're too warm or you're too cold. That'll be then processed somewhere in your brain, which is going to tell various parts of your body to react. And so, for instance, if we're talking about this thermostat example, right? Your thermostat reads the temperature in the room. If it's too hot, it's going to tell your AC to kick on and bring down that temperature. If it's too cold, it's going to tell your heater to warm up the temperature of the house to keep it at 68 or 72 or whatever you keep it at. Basically, you have two, and we talked about this especially a lot with the endocrine system. You often have these two different hormones that are paired together. Think, what is it? Insulin and the glycogen, I believe it is, off the top of my head. But where one will increase the amount of blood sugar and the other one will decrease it. And what that does is it allows you to kind of aim for that perfect midpoint, that homeostasis. So negative feedback loops are primarily there for maintaining that homeostasis, whereas positive feedback loops are there to kind of ramp something up into where it can't be stopped. For instance, probably the best example of this is during childbirth, because you have just very basic things that are going to interact with the cervix, interact with all these different endocrine system relations, and increase the amount of serotonin, as well as, yes.
Just for visual purposes, so positive feedback would just be like a straight increase,
Exactly.
and then a negative would kind of just be.
Exactly. So if you're talking about a positive feedback loop, usually the way the reaction is going to work is it'll be like a perfectly exponentially growing curve. Whereas the negative feedback loop, it'll be kind of like, say, this is the midpoint here. If something happens, it'll come back up and do that, or do that. Where it's kind of always perpetually chasing that middle line. Does that make a little bit more sense? Awesome. Now, another thing I did want to focus on, and this was a question that people kind of had a little bit of struggle with, and it's probably because we went through it somewhat quickly on Monday. There's obviously a big difference between asexual reproduction and sexual reproduction. Asexual reproduction, you're talking about a situation where a single organism on its own is able to produce its own offspring. There's no mating involved, anything like that. Where sexual reproduction, you have to have two different parents. They can be hermaphroditic parents, where both parents are male and female at the exact same time. They can be, single sex parents, all that kind of fun stuff. But ultimately, when it comes to asexual versus sexual reproduction, what's happening here is you have a situation where in asexual, the parent is able to reproduce on its own, and sexual, the parent is having to rely on interacting with another parent. Now, both of these can be advantageous in different environments. If you're in a perpetually stable environment, asexual may be the way to go. But if you're in an environment that changes relatively frequently, like most environments tend to do, sexual reproduction may be better. However, obviously sexual reproduction is a lot more costly. There's a lot more energy that it takes to develop these systems and make it where you're not just budding off from each other. You're actually having to, find ways to, one, be able to mix your genes so it's not just the same exact offspring over and over again, but two, finding a mate, going often through territorial battles, all that kind of fun stuff is what makes sexual reproduction so much more energy costly. So while most of the time when we think of asexual reproduction, it's usually in invertebrates, right? Or bacterial cells, or a lot of fungi or plants will do this. It's not just isolated to those groups. Specifically, there's two groups that I do want to mention: invertebrates, which are your mammals, your birds, your reptiles, your amphibians, your fish. And we'll talk more about those specific groups and how they all break down everything at some point here soon. However, both snakes and lizards can do this through a process called parthenogenesis. Parthenogenesis basically means that this organism is now essentially a clone of itself. It can be both facultative, which means it's the only way it will reproduce. Or sorry, it'll either be obligate, which is either the only way that organism can reproduce, or facultative, which means that that organism can kind of turn it on or turn it off depending on the situation. Very specifically, you have a lot of snakes and lizards that are primarily facultative in this regard, where, say for instance, you're out on your own as a little lizard living in this desert landscape, and you cannot find a mate. Well, what you'll do is you'll go through parthenogenesis, basically clone yourself so that way you can continue, your genes at least can continue living on to that next generation. A lot of snakes can do this, a lot of lizards can do it. It just kind of depends. I'm sure I've probably told this story before. There's a cottonmouth at Zoo Atlanta called Preacher Killer. She was the first cottonmouth ever seen to do this parthenogenic birth, where just one day she had never been around any other cottonmouths her entire life. And then all of a sudden she had like 10 babies. Like 9 of them were dead on arrival, which usually happens with parthenogenesis, and 1 of them has lived. And so that's the animal you'll still see there on display 10, 20 years later. Unfortunately, she passed away. She was like 30 or 40 years old, which is stupid old for a snake. There's another great example of this that happened in green anacondas recently. There's an entire blog where this guy who runs a reptile zoo had one of his green anacondas go through parthenogenesis and had its own offspring. Now, mind you, not all snakes lay eggs. So things like boas, so anacondas, a lot of vipers will just have eggs that develop inside of the female. And there's a lot of specialized terminology for that, and we'll talk about that at some point. And ultimately what happens is usually with facultative parthenogenesis, it's like 9 out of 10 of the individuals are just not healthy. They don't develop right. There's just some sort of weird malformation in the genetics that causes major issues. And oftentimes the offspring that do live aren't always the healthiest either. That green anaconda that I mentioned died a week later. Even though it looked healthy, there was congenital issues with its intestinal organs, and it just died pretty much instantly. So it's not always a perfect solution, but it exists out there. Again, lizards are a great example of this. But we were talking about obligate parthenogenesis. A great example of this is go walk outside tonight where there's a well-lit area, like there's a street lamp or something like that. And usually, at least here in Central Florida, you're going to see a bunch of little lizards scurry along the sides of the walls. These are Indo-Pacific house geckos, Hemidactylus mabui. What's really cool about them is the entire species is female. And what's really neat about this is basically it's made them a perfect invader. That's why you find them everywhere. They're not native to here. They're from, I believe, India or Sri Lanka, one of the two. And what has happened is because they were able to get here and start a population with just like 2 or 3 individuals, they've been able to flourish because there's, they're not reliant on, having a big enough population to worry about genetic loss through sexual reproduction. They're entirely reliant on the safe sexual reproduction. Now, what that means is they're often a lot more vulnerable to threats in their environment, like maybe extreme heat, extreme cold, whatever, but it can completely wipe them out. But at the same time, for whatever reason, because they exist in such high numbers, usually there's enough that leaks squeak by. So I did want to address at least like asexual versus sexual. Some of the organisms that do it, the big thing is when it comes to vertebrates, Just remember, snakes and lizards are the only ones that really do it. Turtles, crocodiles, birds, mammals, all that stuff aren't going to be able to do that. It's just too, for whatever reason, those keep in mind too, right? When you're talking about, come on. This will work. All right. So when you're talking about the vertebrate family tree, right? At the very base of it are things like fish. Let me try to adjust the brightness up a little bit. That made it worse. Is that at least visible? Okay. So what happens is that fish became amphibians. Obviously, some just stayed fish, right? But you have amphibians that we have today. So frogs and salamanders. Whereas this branch broke off and became reptiles isn't really the best way to call it. But for colloquial sake, we'll just call them reptiles. So then you have modern reptiles, which are things like turtles and alligators, lizards, whatever. And those kind of often get branched over here. So turtles, crocs, that'll be our snakes and lizards. Then you have mammals. Then you have dinosaurs, which then became birds. So you can kind of see where basically right here at this line, you have this kind of like what we call the upper level vertebrates. And we'll talk more about this at another point. But basically that line here is where you stop having the ability to have parthenogenesis. All the other groups that are left on that reptile group are, without it. Okay. So hopefully that kind of clears it up a little bit more. But again, when it comes to sexual reproduction, a lot of things do it too. And again, you can keep in mind that whole facultative versus obligate thing. A lot of plants can be both facultative and obligate when it comes to reproduction, because you'll have grasses that can, sexually reproduce using flowers, but they can also just bud off and kind of do their own thing. And that's how you get those big mats of like Bermuda grass or whatever that you're stepping on outside as the front lawn. Those things are based off of budding and kind of spreading out, and not just from constant sexual reproduction happening over and over and over again, to fill each one of those being a different piece of grass, a different individual grass, if you will. But ultimately, most vertebrates are going to just stick to this plant, even if it is a lot more energetically costly. Along those lines, people seem to kind of be confused with the whole indirect versus direct thing. So remember back to, I kind of made fun of this. In Pokemon, we think of evolution, right? Where you're going from the little baby form to the adult form. That's really not called metamorphosis, or sorry, it's not called evolution in the real world, it's called metamorphosis. And what's happening is you have something like a caterpillar that has a complete different body history, how it's going to interact with the environment, and then it becomes something totally different. And oftentimes these adults can have very shortened lives depending on what they're interacting with. For instance, I'm sure y'all have heard all the crazy amount of talk that was about like the 17-year cicadas that everybody was talking about in the summer, right? It's kind of a joke because nothing really happened, at least down here. But what happens is these 17-year cicadas will live as little grubs in the ground for 16 years of their life, just kind of chilling out, and then something innate inside of them, whether that be through hormones or genetics or whatever, tells them, okay, this is the year that I need to emerge or to go into metamorphosis and then emerge out the following year. So anything that's indirect is going to have that metamorphosis step. So that's things like frogs, because you go from a tadpole to a frog, right? They're very, very different. Tadpoles have gills. They are able to handle a lot of things that an adult frog can, because it has legs. It can be out of water, that kind of stuff. But when it comes to direct development, that's going to be more of your classic vertebrates, right? Your turtles, your lizards, your snakes, things that look like the adult from the second that they are created, right? So the second that a snake is, hatched from the egg or born or whatever, it looks like a little juvenile version of, that individual. However, This can have a little bit of a wiggle room here. Obviously, things like birds, things like mammals, we do look a little bit different, but we're not going through a full metamorphosis state where we're changing our entire life history between that juvenile versus the adult form. Ultimately, what's happening is, say for instance, with this turtle, it might have a slight diet shift or something like that, but it still relatively looks like it's adult, right? So their direct developers can be broken into two groups. You have what we call altricial and precocial. And we don't need to get too far into the weeds, but just know that one means that it's more developed when it first emerges, and the other one means that it's less developed. So it's still moving towards that adult body plan, but it's not like it's going from one completely different form to another. If that makes sense. All right. Any other questions? I know somebody mentioned the heart thing. Yes. Okay. Yeah, we can do that. So let me start pulling some of those things up here. A heart would have been 14. Y'all, give me just a second. I'm trying to make this as quick as possible. All right. Okay, so when it comes to the heart, right, there's two atriums and there's two ventricles. You have a right and a left side. Now, atrium, remember, it's like the opening of your house. It's where your blood is coming in. And ultimately, blood is going to move from the left side of your heart to your right side of your heart. So when blood is coming in from all of your other veins that are sitting outside and like it's been exhausted, there's no more oxygen in them, it's going back to the heart, right? It's going to come back in through this left atrium here. That left atrium is then going to pass into the left, sorry, it's backwards on mine. Sorry, the right atrium is then going to pass into the right ventricle. And that right ventricle is going to pump it into the pulmonary artery, which is the only artery that lacks oxygenated red blood cells, right? Because it's going to the lungs, but it's still going away from the heart. It's going to go into this pulmonary artery, go into the lungs, pick up whatever oxygen it needs. Then it's going to come back through the aorta. The aorta, or sorry, back through the pulmonary valve here into the left atrium. That left atrium is going to drop into the left ventricle, which is actually that pumping action there. That's going to pump it back out to the aorta. Ultimately, just remember the big thing here, especially when it comes to questions, is remember right versus left, atrium versus ventricle. Which one's pumping in, which one's receiving, and which direction is it going? Is it going to the lungs or is it going out to the rest of the body? Or coming in from the rest of the body? However you like to. Yes.
Is the heart the first one to be oxygenated after all the blood?
Exactly. So what happens is off of the aorta, there's a secondary artery that comes down here and will actually feed into all the muscle tissue here. I forget the exact name of it off the top of my head, but coronary arteries, yes. So basically what happens is you can see this aorta here, and it comes down, and all these aortic arteries are going to come down and feed all of that muscle tissue that's actually supplying your, that providing your heart the ability to work, right? Yeah. Then monocots versus eudicots. 1 second.
Is the vein coming back from the lungs also called pulmonary vein?
I believe so.
Okay.
Those, like I said, I wouldn't stress too much about it unless it's like a specific thing. Because keep in mind, like if you were taking an anatomy or physiology course, you need to know every single artery and vein that's in there. Here we're just kind of just focus on the big picture stuff like the heart, the aorta, like the very prominent stuff that's incredibly important as far as like being able to function. Oh, where is it? All right. So the big difference between eudicots and dicots are the internal structures of the plant itself. So they're slightly different depending on whether you're talking about the roots or the stems, because obviously the stems are going to have very different patterning that's been present in the roots. But ultimately what happens is in monocots, you're going to have this much more concentrated thing here in the root system, where you basically have like a big concentrated cable fiber sitting in the dead center of the root. Whereas in eudicots, a little bit less. It's not going to be a big concentrated thing with all these little perfect little circles here. It's going to be a little bit more smaller. It's a little bit more broad spectrum, if you will, like where it's kind of spread out throughout the rest of the thing. And notice they're a lot smaller compared to the size of like the flow and the style in those monocots. However, if you look at the, where is it? There we are. So if we're looking at, oh, that's not it. Apologies, y'all. So looking at the actual stems itself between a monocot and a dicot, and a monocot, you're going to have all these little vascular bundles that are kind of spread out throughout the entirety of the stem, versus when you're looking at a eudicote, they're going to be much more kind of collected together on the edges of the stem. Hopefully that makes a little bit more sense. The big thing here with this stuff is make sure you're differentiating roots between stems, and make sure that you remember, okay, in which one, what do they look like? So in a monocot, you're going to have that big central core for the roots, and for the stem, it's going to be a lot more spread out and kind of intermixed like that. But in a eudicote, it's almost opposite, where you have these big central points all the way around the outside. The stem versus in the roots, it's going to be a lot smaller, little tiny connections. Yes. Yeah. So when you're looking at the roots here, these eudicots are going to have a bunch, it's going to be centralized, relatively speaking, but they're going to be a lot smaller, and they're going to be not like, how's the best way to put this? It's not like a bundle of cables together, right? There's not a bunch of just big cables all surrounding the internal. It's like have you guys ever seen like the inside of like a fiberglass cable, where you've got a bunch of like larger circles with a couple smaller circles on the inside? That's what you're going to find in a monocot, versus where a eudicote would be a lot more ubiquitously the same size. That makes a little bit more sense. Yes. Sure. So I don't need to pull up the slide for that one. But when it comes to external versus internal fertilization, remember ultimately what's happening here is you've got some organism that has some sort of way without broadcasting their sperm or whatever out into the environment to inseminate a female, biological female. So in vertebrates, there's a couple different ways things can happen. So now in fish, they're predominantly external. The one group that is typically not are the cartilaginous fishes. At some point when they branched off really early on in the development of vertebrates, they developed something called a clasper, which are two small kind of like fin-shaped appendages near their cloaca that it's usually a pretty quick and easy way to differentiate male versus female when it comes to sharks or rays or what have you. So when it comes to fish, those are pretty much the only ones that are going to be internal. With frogs, they're pretty much all external as well. The female is going to just lay her eggs in the middle of the pond, and the male will go back on top of it and kind of spread sperm on top of it. Kind of gross looking, to be honest. And there's some really fascinating ways they do this. It's kind of gross, but it's kind of cool at the same time. There is one group of frogs, though, called tailed frogs, that can reproduce, or have internal fertilization. I'm not going to have y'all worry about that stuff. Just know that in general, frogs and fish tend to be external. Sharks, things like mammals, obviously, birds tend to be internal. A lot of reptiles or pretty much all reptiles tend to be internal as well. They may not have copulatory organs like a penis or a hemipene, but they may have something what they call cloaca kiss, which is where they basically kind of put their two cloacas together and just let that interchange of genetic material that way. Yes.
So like even if they're like external eggs, they're still being fertilized internally.
Yeah, so things like lizards, birds, all that kind of fun stuff. What happens is all that initial development happens on the inside of the reproductive tract. And then ultimately what will happen is at some point during the development of the egg, it'll calcify the outside of that, and then it'll push the egg out. So things like ball pythons, and this has been really well documented in ball pythons, because people breed them for thousands of dollars. So it's really important to know like ovulation rates and all that kind of fun stuff. But what happens is usually about 15 to 20 days prior to them actually laying the egg is when they're ovulating, and that's when they're going to receive the sperm from the egg. Then they're going to go through a shed, and that kind of gives them a little bit more space to move around in their skin a bit, and that's when they're going to lay their eggs. And it kind of depends. Like obviously, when we think of eggs, we think of like birds, where it's very hard and calcified, right? But a lot of other organisms have almost more of a leathery feel to it. Like and it'll make it actually pretty easy to identify things if you ever see them.
Can you kind of go over the two types of immunity and like the defensive immune system and like the parts in each one?
Yeah. So when it comes to immunity, ultimately the two big picture things you need to think about, you have your humoral immunity and you have your innate immunity. Humoral can also be known as adaptive immunity, but ultimately the big picture item here is the adaptive immunity consists of things that are going to remember and have specialized targeted attacks towards very specific pathogens. Whereas your innate immunity is going to be things that are just like a part of your body. They're just naturally always floating around. Things like a lot of their very basic white blood cells, things like basophils, eosinophils, that kind of stuff. They're there literally just kind of floating around constantly. And if they come into contact with stuff, great. But they're not going to have any differentiated immune response. So they're not going to respond stronger to a bacterial cell that they've seen before. Now your B and T cells are. If they've interacted with that bacteria before and they've built that immunity to it, they're going to have a much stronger reaction, much faster than they would if it was just a natural part of the innate immune system. So basically what happens is, so say for your innate immune system, it's always going to have kind of a nice constant reaction to something. It's not going to be very strong per se. It's not going to be as specialized, but it's always going to have that ability to respond. And that's why there's a lot of organisms outside of vertebrates that just have an innate immune system. And arguably, even a lot of vertebrates just rely on their innate immune system. Pretty much anything below reptiles, it's almost entirely reliant on their innate. Yes, they do have that more evolved system, but they don't rely on it as much because it's a lot more costly. However, when you're talking about your adaptive immune response, yeah, it's kind of slow initially. It'll take a little bit to figure it out, but that second time you're going to have a massive spike and a very quick reaction. The third time you're going to have a very massive spike and a quick reaction. Now, ultimately, if you're exposed to that same antigen over and over and over and over and over and over again, your immune system will know, hey, probably not something I need to keep responding to like this. And it'll slow down that reaction at that point. And that's usually what happens with allergies. And why you may have had an allergy as a kid and now have lost it, because initially your immune system was just really hammering that particular allergen. And now it's like, what the fuck? Why do I care about this? And so it's the big picture here is you've got your adaptive and your innate. The adaptive is going to have much stronger reactions and are going to learn. Your innate is just going to be always there and present. And it's also going to include things that aren't necessarily what you think of as your immune system. Mucus in your nose, all that kind of fun stuff. Yes. Yes, I'm going to probably need to pull up the PowerPoint for that one, but in the meantime, I can take another question while I'm doing that. Yes.
So you were just talking about how our immune system allergies change.
Yeah.
Just with your system's like, oh, I don't need to worry about that. So what about like allergies to things like shellfish or like some people have like other foods? Can that like also increase the recurrence?
It depends, which is kind of the unfortunate part about it. Honestly, I couldn't tell you why specifically, but it just, what ultimately ends up happening is there's just certain ways that certain antigens that are present on, say, the outside of, usually what triggers it, say, for instance, like the shellfish or whatever, there's some sort of protein that's present when you're breaking down that food that gets processed in your stomach or whatnot, that your body will perceive as a threat, and then it reacts to it. And what's weird is it can be changed off of very little things. Like for instance, I always love telling this story. There's something called STARI, which is Southern Tick-borne something illness. And ultimately what happens is if this southern tick bites you and it gets its saliva in you, something about that saliva has an enzyme that is very similar to the proteins that are present in red meat. So all of a sudden, you are no longer able to eat red meat without getting violently ill. And all it is just you got this one random bite from a little Lone Star tick that get maybe that big. And as a result, that whatever enzyme that it had present in its mouth is now triggering your immune system to respond to the same way it would also respond to red meat. It's really kind of a weird phenomenon. And sometimes it's just, particularly with allergens, it can build up over time too. There's some people that are definitely allergic to things like poison ivy and poison oak. But what's happening is over time, they weren't that way to begin with. But now that they've been exposed to it over and over and over and over and over again, and your body starts kind of reacting to it more and more and more, ultimately what happens is it's going to get to the point where it kind of basically gets to where your entire body is going to be covered in massive welts the second you touch it. Whereas maybe the first couple of times that you touched it, you had no reaction. And it just kind of depends. Your immune system is a very weird and complex system. Some days it works like it's supposed to, and other days it just decides to fuck up and do stupid shit. Once. Okay. So I'll answer the muscle fiber stuff. We are kind of getting close on time, so keep that in mind. And honestly, if we run out of, like, if you don't get your question answered here, don't be afraid to just shoot me an email or anything like that. I'm happy to chat after class, whatever. All right, so when it comes to these muscle fibers, ultimately you have this myofibril here, that central thing here. All these myofibrils are going to come together to form that muscle fiber. And these myofibrils, they're segmented in what we call sarcomere. So it's kind of like saying, you know, this long road is the entire myofibril, right? So say highway I-4, right? All the way from Daytona to Tampa. That's all a myofibril. A sarcomere is literally from Altamont Springs to, say, Maitland. It's a subsection of that. And within those myofibrils, you're going to have these different filaments inside of it. You have myofibrils, which are going to ultimately contain these thick filaments, the thin filaments. The thin filament is called actin. The thick filament is going to be myosin. And ultimately what happens is there's a chemical reaction based off ATP where these thick and thin elements are going to bind to each other. It's going to bend these little connections here. And when those connections bend, they're going to move that muscle fiber forward or back. Basically, it's kind of like stretching a rubber band. It doesn't gain or lose the amount of space that it's taking up, the amount of mass it's taking up. It's just changing how it's shaped and how far out stretched it is. And then ultimately these connections are going to break, and those thick and thin filaments are going to come back together the way they were. So in other words, the thick and thin filament are going to bind. ATP is going to branch them out like this. That's going to hold there for a second, and then the ATP is going to break away, and it's going to go back to the way it was. And mind you, remember, these can be very, very minute, detailed stuff. So things like the muscles in your face are going to have very, very small, fine motor movements based off of the size of the myofibrils themselves. Or it can be massive. Like something like the muscles in your calf, for instance, are going to be huge. And so the myofibrils there are going to be a lot bigger and often bunched up in a lot higher densities. And usually when they move, they're going to move much larger distances than, say, something like in your face, which might just go. All right. Any other questions? All right. Give me just a second.